the research that happens in my lab at the University of Georgia, where we study malaria parasites. And shown here are a couple of pictures, images from that was taken in our lab of malaria parasites that are about to invade red blood cells. As you may know, malaria is a very ancient disease. This is a picture of a mosquito trapped in amber from 20 million years ago. And in the midgut of that mosquito, you can sort of see these parasites that are present. In fact, if you think about how much human death has been caused by malaria since the Stone Ages, about half of all human deaths from natural causes, this is excluding all wars and accidents, this has caused half of all deaths since the Stone Ages have been attributed to malaria. It is a global problem. Shown here is actually, I like to start with a picture of the United States from about a century ago. And you can see malaria is present all over the US, shown in highlighted in red here. And in fact, it was only very recently, about 60 or 70 years ago, that the US finally managed to eradicate malaria. This is Georgia down here, and this is where I am currently present. And as you can see, this whole state of Georgia has had a lot of malaria since the beginning. And interestingly, in the very beginning of the state, they had to move the capital of the state around because every time the legislatures would come together to have a Congress, they would fall sick from malaria. Today, malaria is still present all over the world, particularly in sub-Saharan Africa, where it causes most of the deaths, as well as in India and Southeast Asia. And most interestingly, very recently, there were cases of malaria that happened within the US. So after about 60 years of eradicating malaria from the US, it came back. There were about 6 to 10 cases of malaria where the transmission was local. What does that mean? That means that these were not people who traveled out to a malaria endemic region and came back with malaria. These were people who lived in the US, never left the US, but still got malaria. So this raises the urgency of the problem that we are facing. So why has malaria been so hard to eradicate? It stems from the problem of evolution, actually. So if you think of the eukaryotic tree of life, these are animals. This is where we are, and this is where most of biomedical research focuses on, rightly so, because we're interested in how we work and want to solve diseases of humans. This is where plants are present. This is a fairly big evolutionary distance over here. And this in here is where the malaria parasite is located on the evolutionary tree. As you can see, these parasites evolved out of this branch long before plants and animals separated. So their biology is extremely different, this makes it much harder to study these parasites. This biology is reflected in the life cycle of Plasmodium. As I had mentioned or alluded to earlier, the malaria parasite actually is transmitted by mosquitoes. When a mosquito bites the infected humans, it takes up gametocytes, which then fertilize, the male and female fertilize each other to form a zygote, which then moves to the vector's gut, where it forms a oocyst, then develops into sporozoites, which migrate to the salivary glands of mosquitoes. And when this mosquito bites a human, it injects these forms called sporozoites, which then migrate to the human liver, go inside hepatocytes that are liver cells, and then divide. One parasite infecting a hepatocyte divides into about 40,000 daughter cells. It's the largest known expansion in eukaryotes. And interestingly, these forms that develop in the liver are known as merozoites. They cannot reinfect a liver cell. When they break out of this liver cell, they have to go back into a red blood cell. In the red blood cell, they undertake this life cycle where it takes about 48 hours. A single merozoite enters the red blood cell and then divides into about 24 daughter cells, bursts out of the red blood cell, repeats this cycle over and over. And a small percentage of these develop into gametocytes to be taken up by mosquitoes. In our lab, we focus on the red blood cell stages of the parasite growth because this is the stage that causes all the clinical symptoms. Plus, it is fairly straightforward to grow these parasites in the lab because as long as we keep supplying human red blood cells, these parasites will keep growing. So as I mentioned, we are focused on the red blood cell stages. This begins with the process called invasion, where the parasite shown here, this form known as the merozoite, enters a red blood cell. It actively invades a red blood cell. And then within the red blood cell, it develops into what's known as the ring stages, and then a metabolically active trophozoite, where they consume hemoglobin from the red blood cell to generate material to make their own daughter cells. They divide by a process called schizogony, where they divide into about 24 to 48, 24 daughter cells. These parasites then have to get out of the red blood cell. So they break open the red blood cell, rupturing the membrane, and then releasing the merozoites back into the culture. And now these are back into blood. These merozoites now find a new red blood cell, invade, and this whole cycle continues. And this cycle takes about 48 hours, as I mentioned. And in our lab, we are focused on a few different parts of this cycle. First and foremost, we're interested in understanding how the parasite transforms the host red blood cell. So as I mentioned, this is a eukaryotic parasite, so it has its own nucleus, ER, and so on. And it sends proteins into the vacuolar membrane. So it stays within a vacuole inside the red blood cell. These proteins are then sent out into the host red blood cells, converting the host red blood cell from what looks like a normal red blood cell into a very deformed red blood cell. These are what's known as scanning electron micrographs. So they're false colored. What does that mean? It just means that we colored them. They don't actually look red. They are green. But what you can see are these knobs. So the parasite sends out proteins on the host red blood cell membrane that help it interact with the host immune system as well as endothelial cells in the bloodstream. This has very severe consequences for disease. This is how the parasite sticks to endothelial cell walls in our brain, causing cerebral malaria, which is lethal, or in your kidneys, causing kidney dysfunction, or in your lungs, causing lung dysfunction. We're also interested in how the parasite gets out of the host cell. So as I mentioned, it stays inside a vacuole that it forms called the parasitophorous vacuole. But when it has to get out of the host cell, these daughter cells have to break open the vacuole and then the host membrane. And shown here is a live video microscopy from our lab where we labeled the vacuolar membrane with a red fluorescent protein here. And you can sort of see the shadow of the red blood cell outlined here. And these white dots are vesicles that the parasite sends out, which contain proteins that help break down this membrane. And this is what it looks like in real time. It's a very quick process, and you can see these membrane breaking down, the little merozoites are released, and pretty soon the whole red blood cell is destroyed. And this is a primary driver of disease symptoms as well. And finally, we are also interested in how the parasite gets into the host red blood cell. This is, again, a fairly complex process that involves a lot of different signaling pathways, as well as secretion of proteins from the merozoite into the red blood cell. These parasites have these unique organelles called rope trees, which are these club-shaped organelle at the apical end, and all rope tree proteins are secreted into the red blood cell during invasion. And again, this is a very quick process. This parasite cannot live outside a host cell. It has to stay inside a host cell. And this is, again, real-time image. You just follow the white arrow here. You can see the merozoite attaching to the red blood cell and very quickly sinking into the red blood cell. And so we are interested in our lab in understanding how these protein secretion pathways help the parasite live and grow inside human red blood cells. And understanding this process will help us develop better therapeutics as well as interventions against this parasite. The short and quick story I will tell you today is about how the mirazoid goes into the red blood cell. In particular, this process has been well studied because obviously, if you can block the mirazoids either from entering red blood cells, either by vaccinations or with drugs, you can block this entire infection process. It begins when the mirazoid finds a red blood cell, attaches to it, and then it reorients so that its rope trees, shown here in purple, are pointing into the red blood cell. It then forms a pore in the red blood cell membrane, and rope-tree proteins are secreted both into the cytoplasm of the red blood cell as well as the membrane of the red blood cell. These proteins then form what's called a tight junction. They help the mirazoid bind to the red blood cell really well. And then the mirazoid uses its own motility to push itself into the red blood cell. Finally, finishing with this process called echinocytosis, which we don't understand why that happens really. But this process is membrane ruffling. The red blood cell starts looking very spiky, but it resolves itself after a few minutes. During this entire process, an inorganic ion, calcium, is critical, and it plays a key role in signaling to the mirazoid that it's bound to a red blood cell and it's okay to secrete rope trees and start the invasion process. So our lab has been interested in the field in general, has been interested in understanding what are the mediators of this calcium signaling pathways. And so we focused on this one protein called a rope tree neck protein 11 or RON11. It's a transmembrane protein with a calcium binding EF hand domain. And the work I'm telling you about was done by a recent PhD student who graduated from our lab late last year, Dr. David Anaguano. And this protein is present in not just in Plasmodium falciparum, but also in other Plasmodium species like Vivax and Berghia, as well as other apicomplexan parasites such as Toxoplasma and Cryptosporidium. However, the role of this protein in the biology of the parasite was completely unknown. And so we wanted to study this protein, but how do you study gene function in Plasmodium falciparum? It is a haploid genome. That means it only has one copy of every gene. And so if you take away that gene and it's essential for parasite growth, the parasite will just die and you will never get the knockouts. So what we use is something called conditional knockdowns. What this allows us to do, I'll tell you in a bit. So we have very low transfection efficiency. However, I don't know if you've had any talks on CRISPR-Cas9. This is a tool that allows us to surgically insert specific tags into any gene of interest within the genome of Plasmodium falciparum. And so the approach we use is called conditional knockdowns. There are several ways of doing this. All it means is in the presence of some small molecule in culture, the gene function is disrupted, specifically the gene we are studying. It can be done at the DNA level, where we can use enzymes that will cut up DNA, but only in the presence of a small molecule called rapamycin. So you can delete the gene when you add rapamycin, or you can block RNA expression. So in this case, we insert something called aptamers, where we can add a small molecule or remove a small molecule and block translation of the protein, as well as approaches where we degrade the protein specifically. In this case, this tags allows the protein to be sent to the proteasome and degraded specifically. In all these cases, we have to insert specific tags into the gene that we're interested in studying. In the case of RON11, what David did was inserted this aptamer array into the mRNA of RON11 specifically. And what this allows us to do is, in the presence of anhydrous tetracycline, the protein gets translated, or the RNA gets translated, and you get RON11. And when you remove anhydrous tetracycline, you repress translation of this mRNA. And so you block production of the protein. That is shown here in a Western blot assay, where we are looking specifically for RON11, as well as a non-specific control called EF1 alpha. And what you can see is in the presence of anhydrous tetracycline, you get expression of RON11. When we remove anhydrous tetracycline, we delete or remove RON11 from the parasites. But an unrelated protein that we did not tag there's no effect of ATC on the expression of this other protein in Plasmodium. So from now on, I'll just call this plus RON11 and minus RON11. And we take a very simple approach. We just say, okay, if you remove a gene, what happens to the parasite? What processes in the biology of the red blood cell life cycle get disrupted? First and foremost, we wanted to see if RON11 really does localize to the rope tree neck. So as I mentioned, rope trees are these club-shaped organelles at the apical end of merozoites. This is a schizon where the daughter merozoites are still within the red blood cell. And we're using a technique here that's very recent called expansion microscopy. So these parasites are very, very small, and conventional light microscopy does not allow us to resolve rope trees because these are in the nanometer range.